Okay, everyone, to keep up with time, I think it's good that we start. So welcome to this last session of today that is about choosing your source. And the first speaker we have today is Meytal Landau, that is going to talk about microbial and antimicrobial amyloids in infectious and neurodegeneration. So please, Meytal, the stage is yours.
Thank you. Okay. Oh, yeah, it's working. Okay. So, hi, it's nice to be here. I heard people say that it's nice to travel after such a long time. I agree. But I've been traveling a lot in the last year, and also during Corona. I think that our dedication also to travel to synchrotrons, also when viruses hit, it's stronger than the fear of viruses. So I was lucky to travel all the time. And today I want to tell you about our project that is involving amyloid fibrils. And the ones that we are working on are actually fibrils that are involved in infectious diseases. They serve as key virulence determinants in microbes. They can kill our immune cells, and or they can scaffold biofilms. For those of you who are strictly structural biologists, biofilms are agglomeration of bacteria or fungi, and they secrete outside proteins and DNA and whatnot, and they are very, very sticky. They're super stable. If this biofilm adhere to pipes underneath hospitals, you can grow superbugs with all the antibiotics and bacteria of the hospital running by. They can stick to implants. If you have an infection with biofilm, you cannot treat it with any antibiotics. You have to surgically remove it. It's a huge health and environmental problem. And amyloids are involved in scaffolding and giving this biofilm some of their properties. They're not the only ones, but sometimes they are the main ones. And our work on this virulence determinants that forms amyloid, they led us to discover a new group of amyloids that are from the other side, which are antimicrobials. So there are amyloids that are actually killing microbes, and somehow these two opposite worlds coincide together with from structural biology perspective, and I will give you some examples. But those of you who heard about amyloids in the past, it's probably not related to infectious diseases. It's probably related to neurodegenerative diseases, such as Alzheimer's and Parkinson and AS and prion diseases. And in these diseases, you have you see plaques of proteins in brains, and they also have these properties of being super stable, super sticky. The thing is with the neurodegenerative diseases is nobody really knows what is the etiology of the disease. Some of the diseases, most cases are sporadic. We don't know what's the propensity for these diseases is coming from. And we also don't know the mechanism of the disease. The fibrils are now considered to be some kind of an endpoint. The body is trying to get rid of this protein. There is accumulation of fibrils, but probably something else along the way to forming these huge fibrils is killing the cells. But this is not completely clear. And what is very interesting about these diseases is that some of them, like the prion diseases, are transmissible. And it took Stanley Prushner years to convince the medical field that in case of prion diseases, they are not being transmitted by microorganism, but the transmissible agent is a protein. And if you think about it, it's against everything that we know about eating proteins, where our body is supposed to completely digest them. But in the case of prions, and you can see it with other amyloid proteins that are related to Alzheimer's and to Parkinson, our body somehow, they manage to survive the entire GI tract, reach the blood or reach the brain in some point, template a disease and cause a disease. So for structural biologists, think what type of a chemistry you have to have such a stable protein that basically nothing can degrade. So this question was running for many years, and answer at the atomic resolution came at 2005 from the lab of David Eisenberg. So nobody can crystallize big amyloid proteins, still not today. They're not crystallizable. So the approach that the Eisenberg lab took is to pick the backbone of the fibrils, which is supposed to be the structure, the core, you name it, the spine. And they solved the structures of this short peptide, which are crystallizable. And they showed at high resolution a trend that was already known from fibril diffraction that was called the cross beta. And basically, they showed the formation of tightly mated beta sheets with interdigitating side chains that form what they call the steric zipper. Imagine two beta sheets mated together, and the side chains form something like a zipper. And this runs over hundreds of thousands of molecules along the fibril axis. So this illuminated the stability of the amyloid fibril and their self-seeding characteristics. And the Eisenberg lab had solved more than 200 structures of different segments from amyloid proteins. I did my postdoc in the lab. I joined a little bit after they solved this structure. And since then, with solid-state NMR and cryo-EM, now we have structures of larger segments of amyloid, even full-length amyloid extracted from brain, from heart tissues, from liver. So now we know how amyloid look like in their fibril form. And you can see that they have different polymorphs, different structures, but they are all embedded still in the same core of a cross beta. You see beta sheets, you're looking in all of these structures down the fibril axis, and you see the mating of two beta sheets that form this very, very stable core. So this was the dogma of amyloids. They were defined as cross beta forming structures. Now, when we started our work, when I started my own lab, I wanted to run away from amyloids and not compete with David Eisenberg. But I couldn't get any funding for any other projects because nobody believed me that I can do it. And obviously they were kind of right. So I wanted to work on amyloids because I knew how to solve structures on amyloids, how many people can solve structures of amyloids. And I thought, okay, let's go to this cool microbial amyloids that people say that are similar to human amyloids, but there was no high resolution structural information available for any of these microbial amyloids and bacterial amyloids. And I'm really glad that we tried this, we attempted this. So the first structures of a full-length bacterial amyloids we got from a peptide, a cytotoxic peptide that is being secreted by a very pathogenic bacteria, Staphylococcus aureus. I have a personal relationship with this bacterium. I couldn't walk for 2 years after it infected my hip, so it was coincidental, but I'm thinking it's kind of time for revenge. So this peptide is attacking our immune system, killing our T cells. So it's a part of the disease mechanism, the infectious mechanism. And it's a short peptide. It's being secreted as 22 amino acids. And we show that it forms fibrils, it binds the amyloid dye, and it forms crystals that look exactly like the typical Eisenberg amyloid crystals. I've seen dozens of them. You can see how they look like. They look like needles. They're about 1 micron in width. You cannot mount them on regular loops. So it's, doing amyloid crystallography, it's a different ballgame. You need a microfocus beamline. You need to screen dozens of them in order to get a full data set. Sometimes you need to combine 12 data sets in order to solve a structure. It's really kind of a niche of its own. And I was really surprised to see these crystals because the longest that we got in the Eisenberg lab was 8 amino acids. So how come 22 amino acids crystallized? So I was very skeptical, and we fought for this structure for a very long time over many synchrotron trips, both at ASRF at ID223.2 and ID13. We needed microfocus. Eventually, the data came, the one that we published came from APS in the NECAT beamline. And when we solved the structure, we understood why we could crystallize 22 amino acids, because it formed structures made out of amphipathic alpha helices, which we named cross alpha. So cross alpha has a very similar quaternary arrangement to the typical cross beta, the molecules are stuck perpendicular to the fibril axis. You see two mated sheets, but the secondary structure is surprisingly different. And to show that this structure is only not a crystallization artifact, we did fibril diffraction, and we can see the same pattern of a cross. So in a cross beta, you can see a fibril diffraction pattern that matches the distances between the strands with orthogonal distance orthogonal to the distance between the sheets. You can see exactly the same thing in the cross alpha structure, but the distances match exactly to the distances that we see in the crystal structure. So we know that the fibril has the same structure as in the crystal. And if you wonder about the stability, so it's an amphipathic helix, and between the sheets you have a hydrophobic core, kind of similar to a folded regular, three-dimensional structure of a protein. And so you have a hydrophobic core. So on one side that is facing the interface between sheets, it's hydrophobic. And the outer side, which is hydrophilic, has salt bridges and hydrogen bonds between side chains along the helices. So it's stabilized somehow. I can tell you that it's not as stable as the typical cross beta, but it's still very much stable. And it's not just a nice structure. We, because of the reviewer insisted, we kind of hypothesized and then showed that the cross alpha is critical for the toxicity of these peptides against the human cells. So we have a cytotoxic fibrillation that is mediated by cross alpha formation. And just recently, the lab of Engelmann and Conticello solved the cryo-EM structure of the same peptide, showing these beautiful nanotubes. So the units of these mated helical sheets were exactly the same as in the crystal structure, and we were very happy to see that. But with cryo-EM, you can get the supramolecular assembly of a nanotube that you cannot get from a crystal structure. So you can see the advantages of this method, but I was definitely happy to see it not in the crystal form at high resolution. So is it a singular cross alpha structure in the entire universe? Can we find more of this unique feature? And by correlating some facts and trying to find another one, we found this cool antimicrobial peptide from an amphibian, an Australian toadlet, and it also formed a cross alpha structure. And the data collected was in massive 3. This is one of the more powerful beam line for these microcrystals. And you cannot see here very well, but the helices along the sheets are anti-parallel to each other. So think how crazy this is, that also helices, not just beta strands, can form anti-parallel and parallel sheets. So for a structural biologist, I was more interested in the protein science aspect than anything else. But there are also other cool aspects of function. And I was very surprised that the biophysical measurements showed that there can be a transition between cross beta to cross alpha induced by membrane lipids. And we recently managed to solve the cross beta structure using cryo-EM, showing this weird propeller shape of a cross beta. And you think, why does this amphibian need this transition of a secondary structure in a fibril? So what we think is that the cross beta is a non-toxic entity, but is very, very stable and very, very sticky. And when amphibian is jumping to the water and back, there is some sticky antimicrobial protection. And once it sees bacterial cells, the membrane is pushing for helicity, and then you have a toxic form that actually kills the bacteria. But this is a very unique example in general that breaks the dogma that Anfinsen and others taught us about one sequence, one structure. So it's not just polymorphism in the cross beta realm, it's polymorphism of secondary structure of the same sequence in the fibril form, which I searched the PDB is quite unique. So we kind of love these systems. So the next question, how prevalent cross alpha and the secondary structure switch are? And based on what we learned so far, we devised a computational platform in And in a beautiful collaboration that I have in the lab from experimental and computational, we identified by now dozens of antimicrobial and toxic peptides that form cross-beta, mostly cross-beta, but some cross-alpha amyloids. Some have the ability to switch. So some of these peptides, they form these crystals, I don't know how you could, beautiful, ugly, like you choose. None of them diffract over, I don't know, what, 10 synchrotron trips by now. They diffract they don't diffract, or they diffract with really crappy resolution, or with a reasonable resolution, but we cannot still process the data. If somebody wants to help us, please. You know, this is what I did for a living for a very long time. You know, we managed to solve one of these structures with cryo-EM, which is apparently easier for this peptide, not super easy still, because it's helical reconstruction. But you can see another cross-beta structure, but with kinked beta sheets. So this is a feature that was suggested by Eisenberg, actually, to encode for functional and reversible fibril formation. So it's not as stable as non-kinked beta sheets. And basically, with every structure that we solve, I'm showing just you know, the latest one, we discover a new amyloid fold, or call it like formation, that we cannot predict by any method. AlphaFold cannot help us with amyloid fibrils. AlphaFold does not even predict beta amyloid formation. Like, it thinks that everything is helical in life, surprisingly. So it's very important in this case to solve more structure and to broaden our array, because it's the only way that we can generate hypotheses about function, structure-function relationship, about mechanism, about everything. So this is what we majorly do. We do this structure-function relationship. We correlate the biological activity to all the structural properties. There are some structural properties that are crazy, or chemical properties. Some of these amyloids, they're so stable that can survive autoclaving. You boil them, they keep on growing. You put them in formic acid 100%, they don't care. They're really crazy. They are lab killers. You cannot use them on any FPLC. They will kill all of your cuvettes. They will kill all of your tubes. Like, they're really lab killers. It's really hard to work with them in many aspects. So our goals for them is structure-function relation. We want to understand how exactly they are killing cells, when is the fibrillation happens, and how. We want to, like, we have so many different amyloids from different fields. What are the converging and differentiating features? And I would want to focus this project on amyloid prediction, classification, and design. And eventually, I want to reach a point that you will give me a sequence, and I will tell you if it forms an amyloid and what it's doing. So the potential implications for this work is to develop new antimicrobial drugs that will be super stable, for example, for coating of medical devices, food packages, whatnot. I'm not sure if I would give them for people to actually eat. They can be used as materials for novel technologies, which I don't know anything about. Microbes communicate with each other using some of these amyloids. And I didn't talk about this, but there is a connection between the microbes and their amyloids and neurodegenerative diseases. And this is a little bit troubling. Is our microbiome and infections that we have in our life are related to our propensity to have neurodegenerative diseases, and there are a lot of correlative and causality evidence that suggests so. One example is that you can infect Alzheimer mice with some of these amyloids or their bacteria, and you can see that the symptoms are getting worse, and you can see it with different amyloid diseases. We also showed that there are joint inhibitors that we took some inhibitors from clinical trial of Alzheimer's, and they actually reduced biofilm biomass and inhibited the microbial amyloid. So the structural similarity tells you, is there templating? Is it that far-fetched to think that we can get Jakob-Creutzfeldt disease from cows? Can we also get it from a microbe that infects us? And now there are booming papers coming out about the correlation between coronavirus and Alzheimer's. So it's pretty scary, but on the other hand, if we think about it, if we nail down the actual causality at the molecular level, maybe we can start giving people like probiotics that will change their propensity for neurodegenerative diseases. So this is another direction that I would like to pursue. So most of the work that I showed you was done by my Israeli lab, and this is November, so we have a nice weather. The cryo-EM structures were solved by Robert Booker. He was actually mentioned this morning because he left me to join Rigaku, but it was a joint project with Kai Grunewald and Maria Garcia, who just gave a talk in the previous session. It was a CSSB collaboration. Now we have another postdoc, Fabio. Many thanks to ESRF, Beamline Scientists. I spent some of my best hours in my life there, shifting between beams and trying to manually put these crystals on the goniometer. I'm a part of EMBL, and they give me kind of regular access to P14, which is also very helpful, especially with now fibril diffraction that I need to do all the time. And I'm going to be physically moving to a position at the CSSB in Hamburg with DESI and medical school in Hamburg. I will also be part of the EMBL. So the group is just starting in a few months. So we are hiring staff scientists. We are looking for a person with cryo-EM, ET, CLEM, any kind of EM in cryo conditions for a permanent position. And we are postdocs and PhD students. So if one of you is looking and want to move to Hamburg, it's a beautiful city, please write me an email. And thank you for your attention.